Today's topic is a challenging topic. It's not an easy topic. So, like the previous ones we have been trying to cover. So, let's see how we do today. Okay. So, what we will do, we will first define normal acid-base balance. Then we look at how this balance is maintained by chemical buffers, lungs and kidney. Okay. When we talk about kidney, some familiar concepts will, that we already covered will come back to you. Okay. Then towards the end we will look at acid-base disorders, what causes them, and how do you diagnose them based on arterial blood gas analysis. This part, which is oxygen and gas exchange, although it's listed here, but it is not in the lecture today. So I was thinking it could be like what should I say, homework. So if you go through this, maybe on Friday we can have some discussion on this. Not a very difficult topic actually, oxygen and gas exchange. The most important part is how do you interpret arterial blood gas analysis. So we'll try to do that towards the end, okay, and during the tutorial on Friday. Okay, now pH is, when you talk about acid-base homeostasis, you're talking about pH. When you're talking about pH, you're talking about H plus ion concentration, right? Cellular function is very sensitive to H plus concentration. A small change in H plus ion concentration will affect a lot of enzymes and other processes. So for a cell to function well, this concentration has to be maintained within a very narrow range, and you can see the range over here. Okay, normally it is very, very low, so it's about 40 nano equivalents per liter, and very small change is permissible. If there is any more shift in H plus ion concentration, it will affect the cellular function adversely. Okay, because the concentration of H plus is so low, it is customary to express it on a logarithmic scale, which is the pH. So pH basically is an inverse log of H plus concentration, and we will try to look at it in more detail a bit later. Okay, so if you look here, how pH is related to H plus ion concentration. So 10 raised to power minus 6 H plus concentration will be pH 6. So it's the logarithm of the negative logarithm, because this is 10 raised to power minus 6, right? So negative logarithm of the H plus concentration, that is pH. If you look at now H plus concentration, all right, on a scale, so it is very high here and gradually decreasing because the power is in negative, right? So from 10 raised to power 0 all the way to 10 raised to power minus 14. Opposite to H plus is hydroxyl ion or OH minus. So these two will go reciprocally. If one increases, the other decreases. And where do you see they are in equilibrium? Somewhere here in the middle. So at this point, both of them are at same concentration, same amount. This is the neutral pH. We call it the neutral pH. And this neutral pH is at 7. 10 raised to power minus 7 concentration of H plus ions. So this pH is called, the value of pH 7. So this is the neutral pH. If H plus ion concentration increases, the pH value drops, and we say that now the solution is becoming acidic. If H plus ion concentration decreases, okay, the pH increases, and we call that solution alkaline. Everybody clear about this? A very basic concept. Okay. If you look at different body fluids, the value of pH varies among them. The gastric secretions, they contain large amounts of hydrochloric acid, HCl. So the pH is very, very low, less than 1. The pH of the blood or plasma is very close to the neutral, 7.4. Okay. This is the average value of blood pH. The range is 7.35 to 7.45. Anything below 7.35 will be acidosis. Anything above 7.45 will be alkalosis. So this is extreme acidosis. This is extreme alkalosis. Beyond this, life won't be possible. Okay. Pancreatic juice is slightly alkaline, so pH is higher. You see the pH is around 8. And urine is usually acidic because there are a lot of acids which are excreted in the urine. So pH of urine is usually acidic. Now, there are many acids which are produced in our body when metabolic processes take place. You have few examples are there. Carbonic acid. When there is production of carbon dioxide in the mitochondria in all the cells in our body, this CO2 combines with water and it forms carbonic acid. This is the main acid actually produced in our body, okay, the major acid. Then we have lactic acid. If you have anaerobic metabolism, have you done glucose metabolism? Is broken down all the way to pyruvate in aerobic conditions when oxygen is there. When oxygen is less in the environment, it goes all the way to lactate. Okay, so lactate is an acid, lactic acid. You have sulfuric acid, some amino acid metabolism will generate this, and organic phosphates will produce phosphoric acid. So our body is always under this acid load. Acid is being produced in the body. Okay. There is also some bases which are produced in the body, or alkalis which are produced in the body. Small amount of bicarbonate, okay. Ammonia, which is produced by amino acid metabolism, is non-significant because it is quickly converted to urea. So as you can see here, basically our body is always living in the threat of acids rather than alkalis or bases. A very small amount of bases are produced, but large amount of acids are produced every day. Okay. Now blood pH this is the range, as I just told you, 7.35 to 7.45. Anything below 7.35 we classify it as acidemia or acidosis. Above 7.45, alkalosis or alkalemia. So every day this is the rough amount of acid that is produced in the body. Okay. The importance of regulating blood pH is, as I just mentioned to you, the body processes are very sensitive to any change in pH. Although there is a large amount of acids being produced every day, right, the pH of the blood remains very constant within the normal range, which means there are mechanisms which prevent pH change. If you are adding these acids into the blood, the pH of blood should drop very quickly, but there are mechanisms which will prevent that change in pH. Okay. So regulation of pH, there are three major ways how human body prevents any change in pH, resisting any change in pH. Okay. The first one is the chemical buffers which are present in the blood. There are also buffers present within the cells, and I'll give you some examples later, but mainly the buffers which are present in the blood. Any change in pH, they will immediately bring it back to normal. So they are able to act within seconds. If there is any pH change, they will come into action. Second line of defense is lungs. If, for example, blood buffers are unable to bring the pH back to normal, lungs will kick in, respiratory system. This can take from minutes, it will start within minutes, but it may take from minutes to hours for lungs to bring the pH back to normal. If this second line of defense is not successful either, then we go to the third line of defense, which is kidneys. Okay, this mechanism can begin within hours, but sometimes it may take days before it can bring the pH back to normal. So buffers, there are three major buffers. The most important buffer in the blood is bicarbonate buffer. Then we have phosphate buffer, which is very important in the kidneys, in the kidney tubules, where if you remember, after filtration, the filtrate is flowing through the tubules, where reabsorption and secretion occurs. So a lot of buffering systems are operating there as well, and phosphate is the major buffer there. Okay, and also inside the cells, as phosphate is much high in concentration inside the cells, it is important in maintaining the pH in the cells. Proteins are also very important intracellular buffers inside the cells, because you have to maintain the pH in the cell as well, right? Inside the cell, pH maintenance is also very important. So you have buffers which are inside the cells, and you have buffers which are outside the cells in the ECF, mainly the blood. Does it make sense? Okay. Respiratory or lungs, simply by altering the respiratory rate, how much carbon dioxide are we removing from the blood can also affect the blood pH. As I mentioned to you earlier, carbon dioxide mixes with water, forms carbonic acid, which is acid, right? So CO2 is basically acid when you mix it in water. So if lungs are removing more CO2, they are removing acid from the system, from the blood, right? If they are retaining CO2, they are adding, or they are keeping the acid inside the body. Renal mechanisms, there are mainly 3 mechanisms how kidney can maintain pH homeostasis. First one is it can actively get rid of extra H+. It can reabsorb filter bicarbonate, and it can also make new bicarbonate molecules, and we will look at these in more detail in the next few slides. Okay. Now CO2 or carbon dioxide is the central molecule of blood pH regulation. If you want to understand blood pH regulation, you have to understand how CO2 can affect and how it plays a central role. Basically, where it is coming from? It is coming from metabolism. All the cells in our body, they are producing CO2. This CO2 is then taken up by the blood. Okay. In the blood, it will mix with water. It will form carbonic acid. All right. RBCs, what they do, these red blood cells, they help, their main function is oxygen transport. You all know that, right? But they also help in transport of CO2, and they can also generate some bicarbonate and contribute into the blood. Okay. On the other hand, lungs can directly remove CO2, right? So they can actually fine tune the amount of CO2 in the blood. Kidneys, as I mentioned to you earlier, they can actually either get rid of H+, or they can reabsorb bicarbonate and put it back into the blood. So the combination of all these, CO2, carbonic acid, bicarbonate, as I will show you later, will determine the pH of the blood. Okay. Now let's look at chemical buffers first. The chemical buffers, by definition, buffer is a combination of 2 substances, weak acid and their conjugate base. This is a buffer system. Why do you need 2 members in a buffer system? Because you want to resist the pH change in any direction. If pH increases, you should be able to resist it. If pH decreases, you should be able to resist it. So you have 2 members. One will take care if pH decreases, and one will take care if pH increases. So you have both arms of the buffer system. So they are able to either take up or release H+, so that changes in the H+ concentration are minimized. 3 major buffers. First one is bicarbonate, carbonic acid buffer. The second one is phosphate, phosphoric acid buffer, very important in the kidneys and inside the cells. And protein, proteinate buffer, again important inside the cells. In the blood, it is this one, bicarbonate, carbonic acid buffer. Henderson-Hasselbalch equation. It describes the derivation of pH using this constant, okay, the negative log of the acid dissociation constant. in biological and chemical systems. It is useful for estimating the pH of a buffer solution and finding the equilibrium pH in acid-base reactions. I'll show you this in the next few slides. A constant value which is denoted as Ka, association constant. Okay. So, because as we know, H plus concentration is expressed as pH, as log. So, we put log on both sides, log here and log here. Okay. And then we separate the H plus from this equation and we separate this one over here. Okay. And we know that log of H plus concentration is what? Log of H plus concentration is what we are studying today, is pH, right? But not the normal log, it's the inverse log. Inverse log of H plus concentration is called pH. Okay, so if I write it this way, is that okay? Log of H plus concentration, I can replace it with inverse minus pH because it's the negative log, right? Because I took the log here, the pH, so I have to do the same thing over here. This is log of Ka, so I call it minus pKa, inverse. Both sides, because when you do something here, you have to do the same thing on the other side, right? It's an equation. Okay. Now, if I rearrange it, because this is inverse pKa, this is inverse pH, so I swap them, the formula becomes this. So, pH is equal to pKa plus log of, what is this? The conjugate base, and this is the, what is this? HA. Bicarbonate buffer system is composed of two members, carbonic acid, bicarbonate. Now tell me, if I have, for example, this is a beaker and both of them are present in a solution, all right. If I add H plus to this solution, which one of them will be able to resist change in pH? I am adding H plus, these two are there, and I am adding H plus to the solution. Which one of them will bind to H plus? SO3 minus, right? This will bind to H plus, and it won't allow the pH of the solution to drop. It will resist the change in pH, right? Okay. If from this solution, which is in equilibrium, I take out H plus somehow, which one of them is able to give H plus to the solution? This one. It can dissociate, it can give out one H plus to maintain the pH. So, you have these two members of bicarbonate buffer system present in the blood. If there is addition of pH, this one will immediately bind to H plus and resist any change in pH. If somehow H plus is removed from the system, this carbonic acid will dissociate into H plus and add H plus to the system, to the solution. Does that make sense? Okay. Okay, so this is the acid and this is the conjugate base, right? So HA is the acid, A minus is the conjugate base, and this is H plus ion over here, pH. So this is pH, this is the Henderson-Hasselbalch equation. If we want to apply this equation to bicarbonate buffer now, and we know through experimentation that the pK value for bicarbonate buffer is 6.1. It is 6.1. Okay. And conjugate base is bicarbonate, right? And acid is carbonic acid. So we replace them with these here. So this is the equation for bicarbonate buffer. One more change which is made to this equation is that you replace now carbonic acid by CO2, because CO2 is the main molecule. When it is mixed with water, it will form carbonic acid. And this 0.03 is actually derived from the solubility of CO2 in water, okay, and how quickly it can transform into carbonic acid. So normally, okay, we use this to calculate pH. Why? Because we can measure bicarbonate in the blood Number one, we can measure PCO2 in the blood. So these two values we can easily measure in the blood sample of a patient. So we can easily calculate the pH. So this equation, Henderson-Hasselbalch equation, can be used to calculate pH of the blood if you know the values of bicarbonate concentration and PCO2 concentration. Does that make sense? And how it is derived? Okay. Now, one more thing here. What is the, what is pKa? It is the constant. So it shows you that at this value of pH, 6.1, both members of this buffer will be in equal amount. So carbonic acid and bicarb, both members of the buffer system will be in equilibrium at a pH of 6.1. But our blood's pH is what? 7.4. So this buffer is way off the normal value of blood pH. Can anyone guess why? Why not we have 7.4 here? Because 7.4 is the pH of the blood where this buffer should be functioning, right? Any change from 7.4 above or below, it should be able to resist. Are you getting my point? But the pK of this buffer system is 6.1. Any idea? Anyone who has done chemistry before? Organic chemistry, inorganic chemistry? No? Okay. The secret here is, I was telling you earlier, the acids which are produced in our body and the bases produced in our body. And I told you that we are living under a constant threat of acids. So our body is always facing the challenge of acids coming into the blood, not the bases. So the buffer system that you have in the blood should be much more robust in resisting any drop in pH, because acid is always coming into the blood. So you need the bicarbonate component of this buffer to be in much more amount than the carbonic acid component, because that danger is very minimal. But the danger of acidity, because acid is coming, is much more. So you need much more bicarbonate in the blood to resist any addition of H plus that happens to the blood than having bicarbonate. Does that make sense? And the value is like this, 20:1. So bicarbonate is 20 times more than carbonic acid in the blood. Okay? This is because of this pKa of 6.1. If the pKa of bicarb of this buffer was 7.4, then both of them would be 1:1 at 7.4. You don't want that. You want the bicarbonate component to be much more stronger than the carbonic acid component. Do you get this point? Okay. So that's why this buffer is very effective in resisting any change in pH, especially acidosis. Let's put it in context now. Two members, right? Carbonic acid and bicarbonate. Okay. If H plus is added to the system, bicarbonate can combine to it, right? Form carbonic acid and break down into CO2 and water. And we have this bicarbonate reserve. As I told you, the bicarbonate ion is 20 times more than the carbonic acid component of this buffer. So we have this sodium bicarbonate reserve in our body, which is always contributing and adding bicarbonate ions to the blood, so that the buffer is always able to resist any change in pH, especially the drop in pH. Okay. Now the main reason for this buffer system to be the most effective is because of respiratory control of CO2. You see CO2 is listed over here, right? So the lungs can actually fine tune the CO2 in the blood. Is very robust because of two reasons. Number one is pKa is just right, 6.1. It has large amount of bicarbonate reserves, less carbonic acid, and both its members, the carbonic acid can be fine-tuned by lungs. The bicarbonate can be fine-tuned by kidneys. So kidneys and lungs can help maintain this buffer in its most functional form. So now let me show you this in a better picture here. So see, for example, if somehow acid was added into the blood, which means the pH has dropped, H plus is added in the blood. This H plus will form carbonic acid. It will combine to CO2, right? And then the lungs will exhale CO2 out. The carbonic acid level in the blood will drop. Lungs will be able to get rid of the extra acid. Okay. The kidneys can secrete H plus, and kidneys also reabsorb bicarbonate. So they will increase this component of the buffer, which will have more ability to bind to this H plus, become carbonic acid, break down into CO2 and water, and more CO2 will go out. Okay. So if you add H plus into the blood, kidneys will reabsorb more bicarbonate, so it can bind to H plus, form carbonic acid, break down into CO2 and water, and lungs will take their CO2 out. Okay. If the opposite occurs, if there is removal of H plus from the blood, or the pH of the blood has now increased, alkalosis, what will happen now? The respiratory rate will become less. So you will not remove a lot of CO2 from the blood. CO2 will remain in the blood. When CO2 remains in the blood, it will combine with water. It will make carbonic acid. Carbonic acid will break down into H plus and bicarbonate. When you have H plus coming here, the pH will start to go down. Okay. At the same time, the kidneys will now stop reabsorbing bicarbonate. They will actually start secreting bicarbonate. Okay. So this buffer system is physiologically very important because it can be fine-tuned by the lungs and by the kidneys. Now you see the difference between the two members, the bicarbonate over here, carbonic acid over here, right? Bicarbonate can be increased or decreased via the kidney. Carbonic acid can be increased or decreased via the respiratory rate or the lungs. Making sense? Okay. All right. Moving on to the second buffer system, which is called the phosphate buffer system. It has these two members, sodium dihydrogen phosphate and disodium hydrogen phosphate. Okay. So this is the one which will accept acid. So this is the conjugate base and this is the acid. Okay. It is very low in blood. pK is 6.8. It is more important in the kidneys, and I will show you this in the next few slides when we go to the kidney later. Third type of buffer, protein buffer, very important inside the cells. Third buffer system is proteins. Okay, we have proteins in the plasma. Those proteins do not play a major role. So protein buffer is again important inside the cells. Hemoglobin is one good example. How hemoglobin does that? Okay, it mainly buffers the fixed acids beside being involved in transport of blood gases, which you all know it transports oxygen and CO2. And RBCs also generate bicarbonate ions, which is secreted in exchange of a chloride ion. Let me show you here in this picture. Now this is red blood cell. Okay, the carbon dioxide diffuses into the red blood cells. This enzyme is present in the RBCs, carbonic anhydrase. It will form carbonic acid from CO2 and water. This carbonic acid will then immediately dissociate into bicarbonate and H plus. This H plus will bind to hemoglobin. So hemoglobin can bind to this H plus. Okay. The bicarbonate which is produced goes out of the RBCs in exchange for a chloride. This is called chloride shift. So chloride goes into the RBCs, bicarbonate comes out. If you see here now, what hemoglobin is doing, hemoglobin is binding to H plus, so it is sequestering the acid, plus red blood cell is producing bicarbonate. If you look here, again it's the same concept. CO2 generated from the metabolism, it is in the blood, okay. It can combine with water and form carbonic acid, releases H plus, which can bind to, as I showed you previous slide, with hemoglobin. So hemoglobin can bind to this extra H plus, and in the lungs what hemoglobin does, when there is oxygen rich environment, it takes this oxygen, becomes oxyhemoglobin loaded with oxygen. When this happens, hemoglobin gives off H plus, which is what it was carrying. It becomes oxygenated, gives off H plus. This H plus combines with bicarbonate, becomes carbonic acid, breaks down to CO2 and H2O, and this CO2 is now easily exhaled by the lungs. So what basically happened here, if you see here, this CO2 molecule which was generated by metabolism has now reached in the lung through this whole process, and it is exhaled. And in this process, what hemoglobin has done in the tissues, it took H plus, so it resisted change in pH, it took H plus from there, it came to the lungs, it gave off this H plus, which was then excreted as CO2, exhaled as CO2. Does that make sense? Role of red blood cells, okay. Okay, lungs, now this is to show you, this is rate of alveolar ventilation. Normal is 1 over here. This is pH change in the body fluids. So if somebody starts breathing 2 times normal, so this is 1 time, right? 2 times normal. For example, if your breathing rate right now is 20 per minute, you start breathing at 40 per minute, double, what will happen to your pH? 0.2 change, right? Almost 0.2 change in pH, which is a big change for pH. So respiratory rate can actually alter the pH very quickly. And we saw one example, right, where the student was under stress, hyperventilating, and alkalosis occurred, leading to hypocalcemia later on, right? So respiratory rate can affect blood pH very quickly because you are removing CO2 from the system. Reverse is also true. Effect of blood pH on the rate of ventilation. If the pH goes below 7.4, becomes acidic, respiratory rate will increase. All right, role of kidneys now. What kidneys do, as I showed you with the bicarbonate buffer, they actually reabsorb bicarbonate. So they help maintaining the alkali reserve. So they keep that bicarbonate coming back into the blood so that you have enough bicarbonate there to resist any change in pH. Okay? It also excretes or reabsorbs acidic and basic substances depending upon the situation. The pH of the urine is normally acidic. Why the pH of the urine is normally acidic? What reason can you think of? Because our body is producing a lot of acids, right? That's why we have to get rid of those acids in order to maintain the blood pH. So urine is always acidic because you have to get rid of all those extra acids from the system. Okay? But if you see here, the range that the pH of urine can have is large, is huge, 4.5 to 8. In terms of pH, this is a huge range actually. So urine can become extremely acidic, or it can actually become alkaline. If somebody is having alkalosis, then the urine will excrete bicarbonate. So pH of the urine will become alkaline depending upon the situation. But normally in a healthy individual, the pH ranges around 6. Okay? Now what kidneys do? 3 mechanisms kidneys employ. They can either excrete H plus ion, they can reabsorb bicarbonate, or they can even generate new bicarbonate. When you say reabsorb, it means the bicarbonate was in the blood, it got filtered into the tubules, and then it got reabsorbed and went back. It's the same molecule. But kidneys can also make new bicarbonate, which was not there in the blood, but kidneys contributing new molecules of bicarbonate to the blood. So I'll show you some examples of this. This is excretion of H plus. Okay. This is a single tubular cell, okay, in the kidney tubules. This is, I think, distal tubule. Yeah, this is all proximal tubule, loop of Henle, distal tubule. This is happening everywhere. Just a reminder, whatever is over here is going out with the urine, and whatever is here is going back into the blood. So this is reabsorbed here. Okay, ATP is used over here actually. Sodium goes out, which creates an environment in the cell where you have less sodium. So sodium from the tubular fluid will flow in, and one H plus will go out. This H plus will then combine with bicarbonate, carbonic acid, CO2 and water. CO2 will go back in the cell, okay, and a bicarbonate will be reabsorbed. Also there is primary active secretion of H plus. ATP is directly used to excrete H plus into tubular lumen, which will then ultimately go out. Chloride will follow HCl and it will go out in the urine. When this is happening, the pH of urine will be very, very low, right, because acid is going in the urine. So H plus can be excreted either through a primary active mechanism where ATP is used directly here, or through a secondary active mechanism where ATP is used over here and H plus goes out. Two mechanisms for H plus excretion: primary active, secondary active, right? Bicarbonate reabsorption now, all right? Water combining with CO2, carbonic acid, forming bicarbonate and H plus. Now if you see here, this one, these two arms of this reaction, bicarbonate is going this way back into the blood, and H plus is going out on this side. So what is happening here? Bicarbonate is being reabsorbed and H plus is going out. So there is reabsorption of bicarbonate. Okay. Now this is generation of new bicarbonate. In this case, the bicarbonate is there, right? And this bicarbonate has now reached here through all these reactions. It is going back into the blood. So it was there. In this picture now, there is no bicarbonate in the tubular lumen, but there is a bicarbonate going back into the blood. So this is the new bicarbonate. Okay. How it is generated? Through the phosphate buffer. Remember I told you three buffers: bicarbonate buffer, phosphate buffer and protein. Phosphate buffer important in the renal tubules. This is the phosphate buffer. Okay. What it does, it combines with H plus. Okay. This H plus, which is coming from CO2 and water in the cell, forms carbonic acid, breaks down into bicarbonate and H plus. This bicarbonate goes back into the blood. This H plus comes out, combines with phosphate buffer and also ammonia buffer. There are two buffers here, phosphate buffer and ammonia buffer, and it goes out. Okay. So I know it looks very complicated, but remember the key points that I think I've mentioned a few times now. Kidneys can get rid of H plus through active transport, both primary and secondary. They can reabsorb bicarbonate which was filtered at the glomerulus, they can reabsorb it, or they can produce new bicarbonate and send it into the blood. So this slide, this one is an example of bicarbonate reabsorption. See, it was there in the tubular fluid. It has, through these chain of reactions, gone back into the blood. This diagram, you don't see any bicarbonate in the tubular lumen, but there is one bicarbonate going back into the blood. So this is generation of new bicarbonate. Okay. You want to have like a couple of minutes break? It's too heavy, right? Okay, so we'll just break for a while.
No problem.